This is your recording for the Biology EOC review. The EOC is coming up next week. That's May the 9th. And so we're going to review some things. You need to have your review sheet handy to write things down as you go through the review. So I know that you think that this is kind of crazy, like Jeremy thought about his biology teacher. Okay, asking you to remember all this random stuff from a long time ago. But it's not really just random. There's actually a sequence to things, and there's actually a way things fit together. And I tried to arrange the review sheet so that it made sense in terms of talking about the big ideas that biology surrounds. So, yes, you're a poor baby because you have to take this, but it's not the end of the world. I'm really not terribly concerned about you passing it. I think you'll pass it just fine. I want you to do very well on it and make a very high score. So the first strand, the first big idea around which biology is organized is cell structure and function. And the first thing on your review sheet is list the characteristics of prokaryotes and eukaryotes. So just as a review, okay, the prokaryotes have cytoplasm, DNA, ribosomes, a cell membrane, and a cell wall. And eukaryotes have those same things plus other membrane-bound organelles, and the DNA is in a nucleus. Some of them have a cell wall, some of them don't, depending on what kind of cell they are. Big difference between prokaryotes and eukaryotes. Eukaryotes have a nucleus and organelles, and prokaryotes do not. Secondly, we got some words that we need to talk about because there's a lot of biology that's wrapped around or involving these various terms, and these are terms we'll use numerous times, okay? One is homeostasis. Homeostasis is maintaining a stable internal environment, keeping things just the way they're supposed to be, the optimum for the organism to survive and maintain stable environment. Secondly, we have active transport. Active transport is transport into or out of a cell that requires energy. It's not just simple diffusion or osmosis. It's actively requires some input of energy. And there are several different types of transport that we will mention in just a couple of minutes. Passive transport is transport into or out of a cell that does not require energy. So this is going to include osmosis and diffusion, and facilitated diffusion, where the particles diffuse through a membrane protein. Next, we have some solutions, and you need to identify whether the solutions are isotonic, hypertonic, or hypotonic. So on all these pictures, the cell is on the left and the solution is on the right, and we're talking about a solution with sugar molecules in it. Remember that the cell, we want to maintain homeostasis in the cell, so it's best to put the cell in a solution that is isotonic, meaning it has the same concentration of solutes as the cell does. So when you have a cell that's like this one in a solution that has more dissolved stuff outside the cell than inside the cell, that's going to be a hypertonic solution, and water is going to move out of the cell, which means the cell is going to shrink. When you have the same dissolved solutes on both sides, you're going to have an isotonic solution. This is the best possible environment for a cell to be in, because water is still going to move across the membrane, but it's going to move equally in both directions. And then finally, when you have more dissolved things inside the cell than outside the cell, that's going to be a hypotonic solution, and water is going to move into the cell, causing the cell to shrink. Either hypertonic or hypotonic solutions can be very harmful to the cell. They can damage it and sometimes even cause cell death, depending on how strong the solutions are, how long the cell is located there, and so forth. All right, some examples of active and passive transport. We have active transport, including endocytosis, exocytosis, phagocytosis, penocytosis, and ion pumps. Endocytosis and phagocytosis and penocytosis are all the same thing. That is bringing material into the cell by enfolding of the membrane. Exocytosis is the reverse of that. Phagocytosis specifically talks about bringing in, or the cell eating, bringing in solid materials. And penocytosis is talking about bringing in droplets of liquid. The ion pumps are going to pump ions against their concentration gradient. And an example is the sodium-potassium pump, which we talked about. And those also require energy. Passive transport includes diffusion, osmosis, and facilitated diffusion. Remember, diffusion is just movement of particles from a high concentration to a lower concentration. Osmosis is diffusion of water across a membrane. And facilitated diffusion is diffusion through a protein channel in the membrane. And this is for things that are either too highly charged or a little bit too big, but they have special channels that are made for them. Now, there is a video review on active and passive transport. I am not going to play that in this review, but I will post those videos separately. Next, we have the 4 categories of organic molecules. We have carbohydrates, and they include elements carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. The monomer is called a monosaccharide. That means a single sugar or simple sugar. Polymers are polysaccharides. Sometimes you might also say disaccharides are polymers, but a disaccharide is just 2 monosaccharides put together. And the functions of the carbohydrate are for energy and for structures. There are structures like the cell walls that are often made of carbohydrates, and of course all cells use the carbohydrates for energy. The basic structure is shown this way. You're going to see a hexagon or some kind of a polygonal structure, usually with carbons and hydrogens and oxygens attached to the chains. There's also a video called Molecules Gone Wild that talks about all of these. It's done by the same teacher that did the respiration song that we listened to last fall. And again, I will post the videos separately from this presentation. The second category of organic molecules that we listed on the worksheet is lipids. Lipids are also made of carbon, hydrogen, and oxygen, but they're specifically not in a 1 to 2 to 1 ratio. There's usually a lot more carbon and hydrogen and very little oxygen. Usually the oxygen is only found in the carboxyl group on the end of the fatty acids. The monomers are fatty acids and glycerol, and the polymers are lipids or fats or triglycerides, probably the most common polymer of lipids. Their functions are for energy storage, insulation. They make up membranes. Remember, the phospholipids are included in this. And the structures include both saturated and unsaturated fatty acids. Remember that the saturated fatty acids have all single bonds between the carbons in the chain, and the unsaturated have one or more double bonds in the carbon chain that's attached to the carboxyl group. The next category of organic molecules is proteins. Proteins are made of carbon, hydrogen, oxygen, and nitrogen. The nitrogen is found in the amino groups, remember. The monomer is an amino acid, and the polymer is called a polypeptide or a protein. The functions are for structures, enzymes, hormones, and other kinds of things like that in the cell. There are lots of different kinds of proteins found in all kinds of cells. And the structure is going to include an amino group, the NH3 group, the carboxyl group, the acid group, a hydrogen or some other chain, and an R group. That R group is what's different. There are 22 different amino acids, and they all have slightly different R groups. Next, we have a peptide bond. A peptide bond, remember, is a bond between the carboxyl group of one amino acid and the amino group of another. And they go together by dehydration synthesis, like we talked about back at the beginning of the year, where you take an oxygen from one, 2 hydrogens from the other, and produce water here. And then you have a peptide bond between the carboxyl and the amino groups of 2 adjacent amino acids. The next category of organic molecules is nucleic acids, and we'll talk more about them later. Nucleic acids are made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Their monomers are nucleotides, which are made of a sugar, a phosphate, and one of the nitrogen bases. The polymers are DNA and RNA. The functions are to store genetic information and to produce proteins, protein synthesis, and the structure is called a double helix. And this structure here shows you that you've got the nitrogen bases that attach together by hydrogen bonds. You have 2 strands. One goes this direction, one goes this direction. That's the direction in which they're red. You have a sugar phosphate backbone with the nitrogen bases making up the rungs of the ladder. And remember, the double helix means that it's kind of a twisted ladder shape. Next in Strand 1, we have cell structure and function also, and it's talking about respiration and photosynthesis. On your packet, I told you to write the equations for each and circle the reactants and box the products of each one. So here we have photosynthesis equation, which is 6CO2 plus 6H2O plus energy in the form of sunlight produces C6H12O6, that's glucose, plus 6O2. You need to know the balanced formula for each. The respiration equation is pretty much the opposite. We have the glucose plus the oxygen produces carbon dioxide plus water plus energy in the form of ATP. Now if you're going to circle the reactants and box the products, you should be able to draw in a circle to show the things on the left-hand side and a box to show the things on the right side. There is also a video for photosynthesis and respiration that I will post separately from this presentation. Next on your worksheet, it has you make a Venn diagram comparing the characteristics of viruses and cells. So we're going to put the things that they have that are in common, which is DNA, basically, and the things that they have separate from each other. So cells have cytoplasm, a cell membrane, ribosomes, and organelles, and viruses have capsids, and they both have DNA. Some viruses instead of DNA have RNA, but they all have a nucleic acid. There's also a video on viruses that will be posted separately from this presentation. Define and explain the differences between the lytic and lysogenic phases of the virus life cycle. So here we have the different parts. We have the lytic phase, which is when the virus invades the cell or injects its DNA, takes over the cell machinery, replicates viruses, and ruptures the cell to release. This is the active infection. And the lysogenic phase is when the virus invades the cell and then hides in the host genome and becomes lytic at some later time. There's also a video about this, like I said, that will be posted separately from this presentation. Next, in cell structure and function, we have the stages of the cell cycle and the important events in each one. So we have the G1 phase, where the cell grows and undergoes all normal cell activity. The S phase, S for synthesis, where the DNA replication is performed in preparation for cell division. The cell is still doing all its normal functions at the same time. In the G2 phase, the other molecules and organelles that are needed for division are produced while the cell continues to do its normal functions. And then finally the M phase, which is mitosis, and that's division of the nucleus, followed by cytokinesis, which is division of the cell. Next we have the stages of mitosis and the important events of each one. And there's also a video about this that I will post separately. Interphase, which is not mitosis, but the G1, S, and G2 phases of the cell cycle. That's in between. Then we have prophase. In prophase, the chromosomes become visible, the nuclear membrane dissolves, the spindle starts to form, chromosomes attach to the spindle fibers at the centromeres, getting ready to divide. In metaphase, the chromosomes line up in the middle or the center of the cell. In anaphase, the centromeres split. The chromatids, which are now separate chromosomes, are pulled apart by the spindle fibers. And telophase, the chromosomes start to uncoil and new nuclear membranes form, and cytokinesis follows. This is the end of this presentation. This is just about Strand 1, which is cell structure and function, and I will continue on with other videos for the other parts of the review.